Okay. Uh, good, good evening lah. Almost good evening already. Okay. So, in any way, okay, 1.5 hour until 5:30 for our seminar today. Uh, okay, I'll just simply brief what we're going to do today first. Okay, basically, I will not go through those details. Like it is not a like revision for the two chapters in advance lah. Because as you can see, we have a lot of things here. Okay. Yes. Oh, hello, hello. Okay, so I can't really go through everything. Okay, so what I can do here is I will just tell you which part is the important one, and actually we'll focus more on exercise because in the end, practice is the thing that is matter. Okay, so practice matters lah. Okay, so the rest we'll focus more on exercise. So first thing first, what is important here is you have to know all their names, okay, the apparatus. Okay, what is their name? How do you sketch the diagram? Okay, what is the function? Okay, know these things. Second thing is the hazard. Okay, same thing here. Know the detail, know the logo, know the symbol. Okay, the next part here is the type of accident. You have to know we have a few types of accidents here. Okay, so no need to go through the actions. Like actions is more on logic lah. Okay, when you accidentally hurt your eye, you can wash your eye. Hurt your hand, you can wash your hand, and you tell teacher. Okay, so the action is very logic, but you have to know which kind of accident we have here. Okay, for the physical quantities, okay, what you have to know here is why we need SI unit, okay, and what is SI unit, okay, and what are they. So for all these physical quantities, right, okay, you have to know what is their SI unit. Okay, one very common mistakes that always be made by students is here, the temperature. Okay, a lot of people they always get the wrong information that they assume degrees Celsius is what we so call the SI unit for temperature. This is not correct. Okay, the correct one should be Kelvin. Kelvin is the actual SI unit for temperature. Okay, the next one will be here. Okay, we have several common prefix that we are going to use. Mega, kilo, centi, milli, micro, or even nano. Okay, actually, like even deci, nano, and giga lah. Okay, will come out in your exam. Yes. I have teacher. Oh, okay. You got his notes cover and question, but you wrote my name. Oh, okay, okay. Actually, it doesn't matter. The cover is not matter because maybe my cover is already run out. Okay, so they use the other tutors cover and like question, but everything is actually same, right? I believe actually everything is same. Yeah, yeah, everything is same. That is what important lah because the cover is just cover. Okay, so okay, the next part here is you have to know what is their value lah. Okay, basically the standard form you don't need to go through. What you have to know is the value. How many zeros do they have? Okay. Okay, hold on. It's okay, my dad. Spare me like 30 seconds. Okay, sorry, my dad was calling me. Okay. Then the value here, okay, you have to know what is the value, like how many zeros do they have? Okay, and the conversion matters. Okay, how do you convert from the bigger value to smaller value or from the smaller value to the bigger value? Okay, so we are going to go through the practical part, okay, on this page lah. Okay, so the importance here, okay, why do we have why we need to have those what is so-called SI unit here? Okay, to make sure everything is so fair, okay, to make sure we have common language, okay, a lot of different points that can write. Just take 2 points from here. And for this part, you have to know what is their measuring instruments name, the stopwatch, the thermometer, the what is so called? The ammeter, the voltmeter, okay, know their name here. And one more thing you have to know in this page is the parallax error. Okay, what is parallax error? Which you have to get the correct reading from this angle. Okay, so okay, we skip. Okay, for this one, okay, we are going to through this part, okay, in exercise lah. Okay, we are not going to go through explanation from here because explanation is so complicated. Okay, you need some like example, okay, like as a reference. So for the errors here, we have two major errors. One is systematic error, one is random error. Systematic error is basically refers to the measuring instruments issue. Okay, which means the problem is not in you. Okay, the problem is about the measuring instrument. Okay, you got some problem in the measuring instrument. That is why it's so-called systematic error. For random error, this is not systematic error already. This is your error. Okay, you are the one who have issue. Okay, so parallax error because your mistake, right? Okay, your mistake, you are so carelessness. Okay, you're using the wrong technique. Okay, all these things are considered as so-called random error. Okay, so the next part here, okay, estimating area. Okay, please be attentive here. Okay, uh, estimating area, you need to tick on the boxes which covered more than half. Okay, less than half is not considered. Okay, and you calculate how many ticks you have, you make it to cm cube. But one thing here is the question might be very tricky. Somehow you can see 1 cm times 1 cm. If the question shows you like 2 cm times 2 cm, which means one box here would be 4 cm square already, you know. Okay, one box will be showing 4 cm square. So when you times how many ticks here, then you have to times off how many ticks here. For example, 16. Okay, so an example lah. So this is what you have to be careful of. More than half, you take it. Then you have to know how to calculate density. Okay, density formula is what we so-called density equals to mass divided by volume. Okay, you can play around with the formula lah. Okay, the volume goes to here, become multiply, okay, whatever. Then water displacement method is we are using the difference of water volume, okay, to indicate the volume of the solid. Because volume of water in ml is actually equals to cm cube. Okay, so it is actually equals to the volume of water as well. So you can use the the ml to convert into cm cube, okay, and you find out what is the difference between them, and then you know what is the volume of the solid. Okay, so this is what you have to know. 1 ml equals to 1 cm cube. And the variables here, I would suggest you to read through but not memorize. You don't memorize this thing here because memorizing is more on chapter 2. Okay, chapter 2, what you need to memorize. How are you going to observe those cells? Okay, you have to memorize this thing. Second thing, their name, their function. Okay, in exercise, we barely will practice this thing because everything is just copy and paste. Know their function, know their name, know their structure, that's all. Okay, and you have to know what is their differences, what is their similarities. Okay, for unicellular and multicellular, same thing. Know their name because if you attended my class before, right, I did show you what how they look. Okay, so you might be familiar with them already. So all their names, okay, whether they are considered as unicellular or multicellular, okay, you have to know lah. All right, their name. And then one more thing here, euglena is so special. You have to know euglena is so special. Somehow the question will ask you to justify, okay, why euglena is so special. Then you have to explain because euglena itself, it is considered as plant as well as animal. Okay, because can do photosynthesis and moving, just like animal. Okay, then for the type of the type of cell here, same thing. You have to know all their names and all their function. Okay, there's nothing to argue here lah, because you have no choice. Okay, we have no any tricks here. Okay, just know their name and know their function, just like how you know your new friend. Okay, so same goes to the type of cell for plants. Okay, then this one, this part no need to memorize. What you have to know is the theory for this like small particular sentence. Okay, from cell to tissue, tissue to organ, organ to system, in the end, organism. Just know this thing, and but of course, the theory is theory. You have to apply from epithelium cell to epithelium tissue to become your any organ to form into a system. In the end, become an organism. Okay, so this is what you have to know. And then Okay, system in human body. Okay, this part, we have 11 systems. Okay, the name, you have to memorize their names. 11 names here. And if it is possible for you, you have sufficient memory in your brain, all these names, okay, memorize them as well. The name, the function, and the particular organ. If you really cannot, okay, at least know some common one. For example, lung, okay, the stomach, okay, the small intestine, large intestine, those common one, it is a must. Okay. For those like penis, ovary, testes, okay, common one, you have to know. Like for this one, very particular, thyroid, pituitary, like adrenal, pancreas, okay, like it is not so common. Okay, if you really cannot remember, then you give up lah. If you can, everything here you have to remember their name, their function, their organ. Okay, so next part here. Okay, you see I give you a lot of stars here already okay, cell respiration and photosynthesis, their purpose, okay, to produce energy, to make their own food. Okay, and what is the formula here? Okay, what is the formula here? As you can see, we do energy and photosynthesis, they produce oxygen and food, as in glucose. Okay, when the question asks you what is glucose, glucose is the simplest form of sugar. Okay, this is what is so-called glucose lah. So we have four particular factor that is a must for plants to do photosynthesis. So once the plant they got do photosynthesis, we can test it by using starch. Sorry, we can test it by using starch test. Okay, starch test means what? We are going to use iodine to test whether the plant got do photosynthesis or not. If the plant got do photosynthesis, then got starch. If got starch, the iodine will turn to dark blue. From brown color turns to dark blue. You did this experiment before. Yeah. Okay. Exactly right. Exactly what it looks like the in the book, right? Okay. So you have to know what is a factor lah that is a must for the, yeah, okay, for the plants to go through photosynthesis. Okay, you have to know. So if no photosynthesis, then the iodine will not turn color, will not change color, will remain in brown color. Okay, so here the similarity and differences for cellular respiration and photosynthesis. Okay, if you are able to make it by your own, you can just keep one. Okay, you can just compare by your own. Cell respiration, you're producing carbon dioxide, and photosynthesis is consuming carbon dioxide. This one is going to produce energy. This one is going to use energy because you're using light energy, right? Okay. Cell respiration, you require oxygen. Photosynthesis, you are producing oxygen. So you see, we have a lot of differences that you can say here. Okay, but eventually they both, I mean, not every single organisms, they rely on these two things to survive. Okay, why? Complements which they are going to help each other. Okay, so nowadays, not nowadays, for example, like now, okay, we don't have photosynthesis already. We just left cellular respiration. Then all the organisms will die. Same goes to if we have photosynthesis but no cellular respiration. Then the rest will die. So the relationship in between here is complement. Okay, which means they both will need each other to survive. Why? Because cell respiration is producing carbon dioxide and photosynthesis is using carbon dioxide. Okay, photosynthesis is producing oxygen and cell respiration is going to use oxygen plus glucose. Okay, so the relationship here in between is complement lah. This is what you have to know how to explain on this thing. Okay, so just a very, 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 very slightly, okay, go through of the theoretical part, then you have to go through the practical part. As same thing, I'll just show you because I just went through the seminar for Chinese version, Mandarin class lah. So, okay, somehow several examples, several answers are given. I'll just simply explain it to you. Okay, so show you how does the question ask you, because this is the first exam you are going to face in your secondary school, right? Okay, so as you can see, section B and section C, basically the question asks you what is this, what is that. So you have to recognize what is this and what is that. The beaker, conical flask, retort stand, Okay, the what is it called? The test tube. Okay, so tripod stand. We have a lot of different stand here. Okay, know their name. Beaker, conical flask, and retort stand. How to sketch this thing? Okay, the outline drawing is like just like this. Oh, yeah. Okay, very simple sketching. You don't be like sketch out all the details. Okay, we are just going to outline. Okay, not going to do drawing. Okay, not draw in detail. So the retort stand would be something like this. Of course, you have to sketch it thicker. Okay, so I'm trying to summarize. Okay, just like this. This is what is so called the retort stand. How it looks like. Okay, so the next part here will be, sorry, this is not retort stand. I have, eh, I wrote the wrong thing here. It is actually tripod stand because it's got 3 legs. Okay, it is tripod stand. Okay, so somehow the question will ask you, okay, classify them into few categories. Okay, you have to identify by your own. What is the function of these two things? They are going to use to carry, to hold the chemical substance. And this thing, it doesn't hold anything, right? It is used to support. Okay, what it can do is just supporting. So it is just supporting, and it is used to hold chemical substances. So A, B, C. Okay. Somehow, some of my answer in sentences will be shorter than the answer that you have gotten from the notes or from the official website. Okay, because our drive will provide answer to you as well. But the answer from there, okay, will be different from me because my answer is like summarized. I write in most simple languages. Okay, because somehow not everyone they have good English, they have good BM, right? Okay, so I always try to use the most simple English for Form 1. Okay, so question 2 is so easy that we just skip. Okay, question 3. This is what I want to show you. How are you going to calculate this thing? Okay, this is what is so-called micrometer screw gauge. Okay, and if you want to indicate the thickness, which is this thing, basically you are going to spin this part. Okay, once you can't spin anymore, okay, once you can't like turn it anymore, okay, then you start to get the reading. So 2 steps here. The first step will be the big ruler. The second step will be the small ruler. So for the big ruler, then you take the readings from here. This is the big ruler. Okay, so which figure are you going to get? Okay, you're going to take this figure, this figure, so called. Okay, so it is after 2, but it is below. So it's not 3. It is supposed to be 2.5. Yep, 2.5. Okay, so the first reading is 2.5 mm. So for the second reading you have to compare. Okay, compare from the horizontal line here. So what is this reading? You have to know. Right? It is 0.21 mm because it's 20, then 21. So 0.21 mm. Yep, 0.21. Then you just add them up together. You got your answer. Okay, 2.71 mm. This is the basic reading okay, that you can get from micrometer screw gauge. We haven't include the what is called zero error inside. Okay, so 2 steps that can increase the precision of the reading. How are you going to get a more accurate reading? Okay, we have few methods here. The answer is not just here. Any answers that can be accepted will be accepted Okay, so take few measurement. Okay, like if you don't believe your first measurement, then you take the second time. You take third time Okay, until you get the like the correct measurement. Or you can say you take the average value for few measurement. So like you measure a few times, but you take the average value. Also can. Or you avoid the parallax error. Okay, as I said, parallax error, you have to take the reading from your side according to this angle. Okay, it is correct. If you take the reading from this angle or from this angle, it is considered as parallax error, and you're going to get the wrong reading Okay, so you have to avoid the what is it called? Parallax error. Okay. Yep. Difference between zero error and parallax error. Okay, good question. So zero error means your zero itself got error. I'm going to show you the zero error later, very soon. And this is what is so-called parallax error. Okay, if you take the reading from here or from here, it is parallax error, which means you are reading in the wrong way. Okay. But zero error means your instrument itself, the zero got problem. Okay, the zero got problem. I'll show you later. Okay. So this part we skip because like simple question. Okay. We come back to not we come back. Okay, we just start question 6 first. Okay, somehow your question will ask you like very basic science question, which asks you what is science, what is about science. Okay. So you have to know lah, okay, which field, okay, has relate to which part of science. So chemistry has to relate to forensic scientists lah, which means you have to know who is the criminal or who is the one who committed crime in that particular place by using the fingerprint. Okay, so for those biologists, right? Okay, you learn about the flowers, you learn about the animals. Engineer, they have to calculate a lot of things based on physics. Okay, you have to include the temperature factor, include the what is so called the momentum factor, the what is so called the size factor, the matter, the density. This is what is so called physics lah. Okay, then we define science. Okay, when the question asks you how to define science, this one you have no way to comprehend. Okay, what you can do is just memorize the sentence lah. Okay, it is actually a scientific observation. As you can see, every single observation, every single experiment is systematic. So systematic observation of natural phenomenon. Okay, so what are you going to observe? We observe all the natural phenomenon. Okay, all the things that happen in this world, okay, is natural. Okay, all the scientific, all the chemical experiment, everything is natural phenomenon. So we are going to use we are going to use the system to observe them lah, like systematic observation. Okay, this is what is so-called science. Then C, okay, this one we have tolerance on the answer. As you can see in the notes should be 44 cm squared. I mean the answer lah. But from here, what I can based on what I can calculate here is like 46 cm squared. Okay, so it is very depends on like the tolerance answer lah. Somehow your teacher will have a range, maybe 43 cm squared to 47 cm squared. The answer will be accepted. But what I want to emphasize here is you have to see how much the marks is given. If the question is given 2 marks here, okay, and telling you to show the calculation, you don't randomly just write down the answer. Okay, you will got only 1 mark, okay, the so-called PT mark lah, okay, or you will not get any mark from this question. So remember, okay, you write down the value, okay, you write down the calculation, everything inside lah. Okay, so okay, the rest part is not so important. Then we go to the important one. Okay, as you can see, same thing here. Apparatus, the name, okay, how to fill the liquid substances, okay, the also chemical substances, okay, beaker, conical flask, test tube, okay, know their name, it matters. All right. Then here, okay, that's what I say. You have to know the symbol as well. Okay, so somehow this one is very particular because not everyone know what is ethanol. Okay, not everyone know what is ethanol, huh? So ethanol is actually a flammable substance. Okay, so back to here, the notes itself did show you some of the example already. At least you grab some of the example that is very common. Alcohol and ethanol is same thing. Okay, so if you just study everything here, but in the end you don't know it's ethanol, right? You cannot proceed to the question one. So ethanol is actually alcohol, same thing, huh? So alcohol is flammable. So how are you going to heat up alcohol? You cannot use fire to heat, okay, heat up the alcohol. So you have to use water bath, which means you heat up the water and you let the water to heat up the what is so called the the ethanol. Okay. So okay, then we go to here, the symbol. Okay, you have to know how to sketch a symbol, of course. Somehow the question will ask you as well, which one is more suitable to heat up chemical substance, retort stand or tripod stand? Okay, retort stand is something looks like this, and tripod stand is something look like this. Okay, so based on your what? Okay, so based on your logic lah, okay, you see. Okay, of course, this one is more suitable because you have a space here to put the Bunsen burner to heat up something, right? okay, you have less space here to heat up something. So actually tripod stand is more suitable lah. Okay, as you have a space in between here, okay, to heat up the chemical substances. Okay, so what about burette and pipette? Okay, know the difference. So you, as you can see, the question is very likely to ask you what is a common thing, what is a different thing between the method, okay, or between the apparatus. Okay, so burette is this thing, or pipette is this thing. Okay, the difference between them is burette can measure the liquid more accurately. Okay, it's like you measure like according to droplet, so it is more accurate. But for pipette, right, it is basically used to measure in a fixed volume. Okay, fixed amount of liquid, like fixed volume of liquid also can lah. All right. So question 8, okay, about the prefix. Okay, so prefix, okay, this one is very common question as well. Okay, as you can see, you have to know how to convert them lah. Okay, basically you have to look at the value, the prefix. Where the prefix is about like from big value to like small value. Okay, yeah, from big to small, it has to be multiple. Oh, you haven't read finish, here, right? I give you some time. I mean, after you finish, you tell me. Okay, done. So the prefix, right, you have to first analyze whether the value is going from big to small or from small value to big value. Okay, so from big to small, we are going to use multiply. Okay, multiply. So what are you going to multiple? Okay, so you see, first, this thing itself, it is kg. So what we know here is 1 kg is equals to 1000 gram. And what we know again here is 1 gram is equals to 1000 milligram. Okay, so here it is, the milligram. Okay, so you have 3 zeros here. Okay, so 3 zeros here. So it is actually like this. So how are you going to convert it? Okay, multiple ma. So 0.006, you times 1000 times 1000. So what you got, the answer is 6000 milligram. Okay, so same goes to the next part lah. From meter to nanometer. Wow, nano is even smaller. So from big to small. Okay, so we have another method here. If you want to use this method, we have another method like quicker method. So you know nanometer is like nine zeros, right? Okay, nanometer is like 9 zeros. So now we are going from big to small, okay, multiple. So you're going to be multiplying ma. So how many zeros? Okay, I rewrite again. Okay, 0.000000009. Okay, so multiply means you are going to push the decimal point for 9 times. 1, yeah. 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, multiple. You push back the decimal point for 9 times. So you got 9 nanometer. Okay, 9 nanometer. This is your answer. So same goes to here. Okay, the next part is different. It is changing from small value to big value. From small value to big value, what we are going to use is division lah, or like divide lor. Okay, so when we talk about multiple, it's like pushing the decimal point to behind according to the zeros. Then divide is different, vice versa. You are pulling back the decimal point front. Okay, you're pushing the decimal point front. How many times? Depends on how many zeros you have. So this one is M, but it is different. Okay, small letter M is milli. Big letter M is mega. So mega, we have 6 zeros here. Okay, 6 zeros here. So we push it for 6 times. 1, 2, 3, 4, 5, 6. So the answer is 7 mega Kelvin. 7 mega Kelvin. K equals to Kelvin. Okay, so the last one is easy lah. 0.3 meter, you have to convert to cm. same thing here. Okay, it is from big to small. So big to small, centi is 2 zeros. Okay, so 30 cm. Okay, so this one is very easy lah. Okay, so what you have to do is the technique for this part only, whether it is from big to small or from small to big. Of course, okay, you cannot simply just write in this way, huh? The calculation, you still have to write it out. Okay, you have to still write out something like this. Okay, but the way to help you to calculate it an easier way is this thing. Okay, if you don't have the steps, right, show your working. No working, no marks. Okay, this is what you have to be careful. So B, Miss Alia, okay, she wants to bake 3 cakes. Okay, so each cakes you have 350 grams of flour. How many, okay, flour is going to use in kg? So you do the calculation. Okay, you times 3, and what do you have is in gram. You convert into kg, divide 1000, 1.05 kg. Okay, simple as ABC. Okay, C. So now she's going to make a dress. Okay, the shopkeeper, okay, she told that she's going to use 4 arms length to as a measure, she's using the arm length to measure. Okay, so in your opinion, okay, can she get the required length of cloth? Okay, so asking you something like this. So actually, no. As we said, this is why SI unit has to be like the reason of existence lah. Okay, no SI unit, then how are you going to measure? Because why? Everyone has different arm length. Okay, so the measurement itself in the end is definitely inaccurate. Okay, so that's why cannot. All right. So 2, suggest a better way. Okay, better way to measure the length of the cloth. Okay, so we can use meter ruler. Okay, actually the answer here can be only accepted is only meter ruler. You cannot say ruler, huh? Ruler you have to measure a lot of different times and it might be inaccurate as well. So you have to make sure it is meter ruler. Why? Because it is long enough and at the same time it is standard SI unit. So we have common language here lah. Okay, so analyze the question. 2 marks. How are you going to take 2 marks? One is the better way. Better way, you got 1 mark. Okay, and one advantage is the second mark. Okay, so same goes to here. In your opinion, can you get the required length of the cloth? Cannot. Cannot stands for 1 mark. Then the explanation stands for another 1 mark. So you have to know how to analyze the question. Okay, like you don't simply tikam the marks here. Okay. Then, okay, the one who asked me, right, how do you differentiate zero error and parallax error? Just now I showed you what is parallax error, right? Okay, so this is what is so-called zero error. As you can see, where is it? As you can see, the zero here, okay, same thing here lah. It is a micrometer screw gauge as well. But the zero error means here, without anything, without clipping anything, okay, you already got some problem. Why? The zero here and the line here is not tally. Okay, it doesn't match. Okay, so in here you have difference. So the difference indicate what? Okay, when your zero is above the line here, it means you are going to be less calculated. Okay, you are going to be less calculated. How much you are going to be less calculated? This part lah. Okay, so what you are going to do? You are going to add back. Okay, you are going to add back the difference here. So when we're talking about adding back the difference here, what you can do is, it is so-called negative zero error. Okay, I know you have some question here. Hey, why you're feeling so confused? Why you say add back? Then why it is negative? Okay, but hold on. Okay, you hold on first. Okay, negative zero error, negative. But what is the value? So the value would be the difference lah. 1, 2, 3, 0.03 mm. Okay, but why? Okay, first thing first, when you talk about why, when you have to know why, okay, you have to know what is the formula. If you want to take the actual reading, right? The actual reading, because your zero got some problem already, if you want to get the actual reading, you have to take the initial reading, which is this thing, to minus out the zero error. So when you're talking about minus out, so minus, okay, negative, negative become positive. So in this case, you are adding back. In fact, actually, you are adding back the zero error. Okay, because you less calculate this part already, so you add back, lor. Okay, so with a wire, okay, same thing happens here, lah. So you see the big ruler and the small ruler. So for the big ruler itself, okay, 5, 6, 7, 8. Okay, but we are not going to take 8 because here we have something at the bottom. Okay, so it's 0.5. So the first measurement would be like the big ruler. Okay, the big ruler will be 8.5 mm. Okay, 8.5 mm. So plus, okay, the second ruler, same thing happens here. We take back the indicator line. Okay, so 1, 1, 2. Okay, so it is like 0.37 mm. Okay, so this is your second ruler. Okay, then you add them up together, you do your calculation. So name the type of error. It is what is so-called negative zero error. If you didn't include the word negative, you still got the mark. So play safe lah. If you want to play safe, you just write zero error. But you have to know it is negative. Okay, state the value. The value here is negative 0.03 mm. Okay, so how can the what we so-called how can the error can be avoided? Okay, if you want to avoid the error, right, which means if you want to take out the error here, what you can do here is you minus out the zero error, which you minus the zero error, or you subtract the zero error. Okay, the noun here doesn't matter as long as you bring out the correct meaning. Okay, so BM has been given here lah. Okay, so what is the thickness of the wire with the error? So with the error means this thing lah. So you add them up together, it would be like 8.87 lah, mm. Okay, 8.87 mm. So last question, where the question asks you about how are you going to determine, okay, the actual thickness. So as I said, okay, now you are going to, because what? Because of you less calculate, so it is negative zero error. So what you want to add it back, okay, so you have to add it back. So how are you going to make it to add it back? Okay, negative zero error. Okay, we are going to use negative zero error. So minus, minus. So negative, negative become positive. So in the end, the actual reading is 8.90 mm. Okay, so you can see like one calculation from part B, you have 1, 2, 3, 4, 5, 6, 6 marks from one calculation. So the deal is very worth lah. Okay, obviously. So you better don't throw away the marks. Okay, so done already, right? Okay, done. Then I pass to the next part. No chat. Okay, good. Okay, so okay, talking about question 10. Okay, again the three functions here. Okay, used to measure about what thing. Okay, this one is can measure up to like what thing? Done. Okay, so this one is very easy lah. Just use to measure the length. Okay, so measure the diameter. Measure the inner diameter. Okay, measure the size also can. Okay, so what about this one? Okay, then the question C is asking another thing. Because B is quite easy, so I just skip. Okay, because you have a lot of things to measure. So C, which instrument is most sensitive? Sensitive means what? Sensitive means more accurate. Okay, more detailed. So give a reason. Actually, we say micrometer screw gauge is the most sensitive one. Okay, in here, okay, even though you know it is micrometer screw gauge, okay, however, you don't be like so randomly to write down the micrometer screw gauge in here lah. Okay, why? Because somehow if you accidentally, okay, you write the wrong name already. Okay, maybe I assume you know lah, but if of course if 100% you sure the answer is correct, you can write. But what if just in case your spelling got problem, who knows? Okay, so if you want to play safe, when the question asks you which measuring instrument, right, you can actually write Z. Okay, why? Because it can measure up to 0.01 mm. This is what it can do. Okay, so then give another reason lah. Okay, why you say this thing can help you to get more sensitive? Because it will able to know small changes, or it will allow to measure the small changes also can. Okay, so 3 marks. Which one is 1 mark? Reason is 1 mark. you further elaborate why you say so is more sensitive is the third marks. Can I write diagram Z? Diagram Z. Cannot, because you don't have diagram Z here. You only got diagram 2. Okay, so what you can just write is Z lah, because the measuring instruments here like Z ma. If you write diagram, right, I afraid the teacher will be so strict lah, then they don't give you the mark. Okay. Then okay, same thing here, but this thing is different. Okay, different story already. Okay, now here we are talking about vernier caliper as well as the micrometer screw gauge. So this is the vernier caliper. Okay, this is the micrometer screw gauge. So how are you going to measure this thing? First thing first, as same thing lah. You have to measure whether it is positive zero error or negative zero error. Okay, so this thing now you see lor, your big ruler is always the above one. This is your big ruler. This is your small ruler. So your small ruler is behind your big ruler. Not behind, sorry, in front of your big ruler. Okay, in front of your big ruler. So means what? Okay, means upon measuring, right, in the end, you are going to measure more. You are going to measure extra value based on the difference here. So you are going to measure extra stuff. So what you are going to do? You need to minus out. Okay, when you want to minus out, it is considered as positive zero error. But how are you going to get the figure there? Okay, you need to find two completely matched line. Okay, between these two rulers. Okay, so which one is completely matched between each other there? This one lah. So the figure here will be 3. Okay, so actually you can write in two ways lah. Either 0.3 0.3 cm. Or 0.03 mm. Okay, so in either way lah, you can write in either way. Hold on. Oh, sorry, my SI unit got problem. It is actually back to the same thing. 0.3 mm. Okay, in this way, only correct. Okay, you can write in either way. Okay, either way also can, but I prefer you to write cm lah, because cm is not easy to get wrong. Because if you take the calculation here, right, the big ruler, always use cm1. Okay, so if we are going to talk with an object, same thing happens here. You look at the small zero, the small ruler 0, it is pointing here. So this is the figure that we want. Okay, so basically the big ruler here will be 1 cm, and the small ruler, what about the small ruler? Same thing lah. When you're talking about small ruler, you take the completely matched line. So which one is completely matched? This one, no? The 6th one. Okay, so 1 cm plus 0.6 mm. Okay, so total will be 1.06 cm, because you have to convert this thing into cm. Okay, 1.06 cm. Okay, so in the end, if you want to get actual reading, as I said, you minus out, you deduct, okay, the zero error. So since it is positive, you write down positive lah. So negative, positive become negative. So 1.06 minus 0.03. So the answer will be 1.03 cm. This is your actual reading for the vernier caliper. So the only thing that confuses you, I believe, is how do you get the positive zero error and negative zero error lah. So you just identify, you just examine whether it is considered as you calculate more, you measure extra, or you measure less. If you measure extra, you minus out lor. Okay, if you measure more, if you measure less, you add back lah. Okay, if you measure more, you minus out lor. Okay, the relationship here is just like this. Okay, same goes to your micrometer screw gauge. Okay, so you see this part here, okay, when you calculate, right, you calculate less already. Okay, you calculate less already. When you calculate less, you have to add back. So it is considered as negative zero error. So how are you going to get 0.02? Look at the line. What is the difference here? 0.02. Then you add one negative in front lah. This is your negative zero error. Then when it goes to the calculation, okay, the reading here, how are you going to get the reading? Okay, same thing here lah. You take the first ruler only until the second ruler. The first ruler 0, 1, 2. So you have 2. Okay, but don't forget here, you have something here as well. Okay, so it is 2.5. Okay, 2.5 mm plus. Okay, what about this one? You look at this line, no? What is the value of this line? 35, 36, 37, 38. So 0.38 mm. So total is 2.88 mm. So bring back to the formula. You minus out, minus, minus, negative, negative, positive. Okay, so become you add back the one that you miscalculated. Okay, you less calculated that thing. You add it back, it become 2.90 mm. Okay, so this is how you get the actual reading lah, as we said. Okay, then okay, determine the type of error. The type of error is actually zero error. Okay, why we have the cause of the error? Okay, because what? Because the measuring tool got issue, got problem. Okay, not your issue, not your fault. Zero error is actually a systematic error, not your error. So suggest your way to overcome the error. How are you going to overcome the to solve the error of zero error? To minus law, you minus out, you deduct out, you subtract out. The word here you bring out the correct meaning is fair enough already. The word itself doesn't matter. Okay, you just take out the zero error. So this is how you get the correct reading lah. Okay, so for anyone, if you feel like still confusing, right? You can speak up one. Okay, just DM me or whatever. I will be like explain again to you lah. Because what I want to do is make sure you guys know how to deal with this question. Okay. So question 11 is about water displacement method. So know this thing, okay, their name. So you put water here, you put something inside to measure the difference between the volume. Okay, then that is what is so-called water displacement method. Kaedah sesaran air. Okay, yes. Can I see D? Last question. Sure, sure, sure. Here. Done. Okay. So, okay, water displacement method lah. Know the name here. Okay. So, which scientific principle? Okay, the principle we are going to use is volume of solid and water can be measured at the same time lah. Okay, as we said, the ml is equals to cm cube. So this thing is the principle that we are going to use. Okay, so when the question asks you to calculate the volume of the cork, right? Okay, the cork means this thing lah. Okay, very simple. Just use the difference between these two things. Because why? The 42 ml consists of water and cork as, itself at the same time. So you just take these two things and you just find out their difference, and that would be the volume of your cork lah. Okay, so 42 minus 39, you got 3. So 3 ml is equals to 3 cm cube. Okay, that's all. And part B, okay, part B calculation, I leave it to you because like I don't have calculator with me lah. Okay, so I mean the calculation is just like substituting the what is it called? The figure inside. But what I want to bring out the message is you have to know what is the formula here. Even the question, right? They don't give you formula. Normally they won't give you formula one. Okay, most of the school they don't give you formula. But if the question is giving you something like this, actually this thing is a formula as well. Why? Gram indicates mass. This line indicates divide. Cm cube indicates volume. So already given you density is equals to gram divided by volume. Okay, so just do your calculation lah. But what you have to be careful is how do you get these two things? So you just play around with the formula lah. Okay, D equals to M divided by V. So V and D can switch places. Mathematics. Okay, so M you can just put the D over opposite side. From divide you become multiple. So M equals to V times D. Okay, so from one formula you can you can like expand into two different formula one lah. Okay, just changing their position and find out the answer. Okay, so no big issue, huh? Okay, when the question asks you to sketch, okay, to label the observation here, okay, the lowest density should be the highest one, highest position. So plastic cube should be the highest one. Okay, the one with highest density, the iron ball should be sink at the bottom of the water. Okay, because why? Highest density, higher density in here. So inference, when the question is asking you to give inference, right? Okay, what is inference? Inference means reason. Okay, but this reason, you don't need to do explanation. You just give one reason is fair enough already. So we have formula to answer this kind of question. When the question asks you about inference, okay, we have formula to answer one. Every single inference shares the same formula. So formula is what? The observation is happening because of your explanation. Okay. The explanation here is actually reason lah. Okay. So for example, I use this question as an example lah. Okay. So your observation is what? Your observation can be two things. Plastic cube floats on the surface of water, or the iron ball sinks in the bottom of the water. Okay. So I just take one. I take iron ball. Okay. So you can say iron ball sinks in the bottom of water because of highest density or higher density than water. Or you want to say if plastic cube floats on the surface of water, okay, it is because of it has lower density compared to water. Okay, just write in this way, you got the 1 mark already. So quite easy lah because you have formula. Okay, so we go to the next part. Okay, which should be the next chapter already. Okay, chapter 2. Chapter 2, hold on. Chapter 2 basically is focusing more on memorizing. Okay, which you need to put a lot of effort in chapter 2 to memorize. I don't bluff you. Okay, you have to memorize their name, their function, okay, their organ, okay, such things. So the front part is like true and false. I skip. Basically, we do the writing part. Okay, starting from question 6. Okay, question 6, showing you the cell division of an organism. Okay, which from one parent cell you divide into two parent cell. The question asks you, what is cell division? Okay, very easy, very easy. Cell division is actually one cell divide into two cell. So one cell become two cell. This is what we so called cell division. Okay, so what is the importance here for the cell division to living things? Okay, what is the cell division? Living things, we need this for growth. For growth. We have a lot of answers here. Also, you can say to repair the damaged cell. Also can to repair the damaged tissue. Also can. We have a lot of different answers that can be accepted here. Okay, so one difference between cell division one difference of cell division between the organism in the diagram and the chick cell of human. So chick cell of human, okay, and this thing, okay, what is the difference between them? So our chick cell, okay, when you like damage the chick cell, you're going to produce a new chick cell. But for this one, right, okay, basically, no, not this thing. Okay, actually, human itself, okay, we don't repair cell. Okay, we just make new cell because our body is so big, so we have to make different cell, new cell every day. Okay, that's why you need energy. But for chick cell, okay, chick cell of human, right? Okay, what chick cell do? They don't produce new cell. If chick cell produce new cell, you become tumor already. Tumor, aka cancer. So no tumor. Okay, not going to make new cell. Your chick cell is going to repair damaged cell only. Okay, your chick cell got damaged, then it repair itself, lor. Okay, so it has nothing to do with this thing. Okay, this is not about repairing something. This is about doing new thing, creating new thing. All right. So B, you see, asking you what is this and what is that again. So this one you try to go through. Okay. P, what is P? Okay. Basically, it is pointing about these two things. Okay. The two thing with black dots. Okay. Basically, if you attended my class, I did ask you guys to practice what is this. What is this, right? Yeah. Okay. Good try. Good try. Nucleus. Okay. I write slightly further a bit, okay, for BM version because I'm so lazy to change the name already. Okay, yes, very good. Okay, the second one would be the cytoplasm. Okay, BM is what is so called cytoplasma. Okay, so that's why I asked you guys to know their name, know their structure. Okay, what is this? What about this thing? This is what is so-called mitochondria. As I said, once you see something like zigzag shape, okay, in the spherical shape, right? Okay, it is considered as mitochondria. So and this one would be the sorry, the small black dots would be the chloroplast. Okay, cell wall, cell membrane. So know the things lah. Okay, know these things. Okay, so this one is what is so-called vacuole. Uh, so state three differences between animal cell and plant cell. So you see, I give you the hints already. Your exam is very, very likely to ask what is this, what is that, what is the difference, what is the common characteristics. So you brainstorm lah. Okay, animal cell and plant cell. So you say plant cell, plant has cell wall. Okay, and animal cell don't have. Okay, hold on, I write in short because writing in sentence is like too time consuming already. Okay. So other than cell wall, what you can say? Okay, we can say chloroplast. Okay. Okay, the third point can be the size of vacuole. Okay. Uh okay, this is what we so called for animal lah. Okay, animal cell usually they don't have vacuole one. Even they do, okay, so they will have very, very small vacuole and scattered one lah. Okay. But actually, you can just simply write until here only. Animal usually absent, full stop. Okay, I'm just giving you something extra lah. Okay, so PM, tumbuhan. Okay, okay. So basically, consuming some time. It's okay. So basically, it happens in this way. Okay, the question is quite likely to ask you something like this: the differences from this one and for that one. So you have to get used with the pattern in the question, lor. Okay, so question 7 is what I said. You have to know, okay, what is the steps that is showing here. Okay, like how are you going to observe the microorganisms or the cell? Okay, so first thing first, you take the cell. Second thing, you have to put the solution here. Okay, in for human cell, we are going to use methylene blue solution. For plant cell, we are going to use iodine. Okay, so you cover it, you absorb the excessive liquid that coming out, and you clip it, you observe it through the microscope. Okay, so the steps here you write down by your own lah. The answer is already in front already. Okay, I'm not going to waste time to write down every words here. So just telling you that the step A itself, you are going to take human chick cell and place it on the piece of glass. Okay, so step B, you are going to use keyword here. Solution X is what is so-called methylene blue solution. Okay, why to do so? To dye the chick cell. Okay, dye the chick cell means give the chick cell some color. Okay, so after that, step C, you cover the glass and you absorb the excessive water, place it in the microscope to observe. Okay, so that's all. So draw and label the structure of a cell observed under light microscope. Okay, so this is what we so-called chick cell. Human. Human is not plant, right? Okay, so sketch it out, 3 marks. Okay, and you have like mitochondria. Then you have nucleus. Okay, basically that's all. Okay, just sketch something like this. Just 4 things inside, right? Okay, and label. So draw and label. If you draw, you didn't label, maybe the teacher will give you one PT mark only. Okay, if you do label, then okay lah, cell membrane, your nucleus, this is your cytoplasm, and mitochondria. Don't forget the mitochondria is used to produce energy. Okay, what is the purpose of adding solution X in step B? As I said, why do you add this thing inside? Okay, to dye the cell. Okay, untuk mewarnakan sel itu. You are going to give the cell some color lah. Okay, otherwise it is like slightly transparent and you can't even see any single thing. And then that is not good lah. Okay, so now you have a muscle cell. State one function of the muscle cell. Muscle cell, what is the muscle cell used for? Okay, it is used to enable movement lah. So you see, no muscle you cannot move mah. Very common, right? Okay, so now you have a section through leaf. Okay, so upper epidermis, lower epidermis, palisade cell, and of course you have stoma gut cell. So what is cell Z? Cell Z is what we so call the gut cell. If the Z itself is pointing the hole, okay, then the hole is stoma. So, but it is not. Okay, it is pointing about the meat beside the hole. So it is considered as what? Okay, the gut cell. Okay, what is the function of gut cell? Gut cell is controlling the hole to open and close lah. Okay, it controls open and close of stoma. So you see, it's not tough at all actually. Untuk stoma. Okay, so somehow the question is like you can see those questions that is not going to memorize is very easy, but if you don't memorize what is that thing, you can't even answer the question. Okay, so memorizing the name of those part is matter. Am I tired? No lah, I'm not tired. I'm still very energetic. Okay, can then we go to next part. Okay, again, but we don't do like repeat lah, double job already. Okay, so know their name, or you am. No, you am. You are. You are tired. It's okay. Just listen. Okay, so know their name. Okay, muscle cell, and this is your nerve cell. Okay, and tell them. Okay, tell the question. Where does the system comes from? Okay, which part you can found this cell in the system? So which system? Okay, cell K, nerve. So nervous system. Okay, system saraf lah, BM. Okay, so example of organ. Which organ do you have inside? Your brain, law? Or you want to write a spinal cord also can. Okay, that's why I say know at least the basic, okay, the basic organ in each system. Okay, so cell of the organism, as I said, okay, we don't simply memorize. You have to know, okay, from which part to which part, from cell to tissue, from tissue to organ to system to organism. Okay, so what is the function of system V? Okay, what is the importance and what is the system name? Okay, L is muscle cell or muscle tissue. Muscle cell, because it is showing cell. Okay, it's asking about name the cells, right? So you write cells. Okay, if the question is asking you to write down the tissue, then you write tissue lor. But very less likely lah. Okay, the question usually wants you to write cell one. Okay, muscle cell. Okay, so system V, what is it? Okay, it is your digestion. Eh, what am I for? Your digestive system. Okay, digestive system. Yes. Yeah, digestive system. Okay, what is the important here? Okay, it can, you don't be like playing around with your words, it can help us to digest food. I mean, can be accepted lah. Okay, but try to avoid something like this because if your teacher is so strict, right? The teacher can say digestive system, it helps to digest. Then respiratory system, it helps you to do respiration. It helps you to do respiration. Full stop. It doesn't, it doesn't give any point. Somehow some strict teacher is so strict until this kind of answer, she won't accept one. Okay, so we write down in detail. It helps to break down food. Break down food means digesting the food lah. Okay, and absorb nutrition. Not just digesting the food, as well as absorption of the nutrition. Also got. Okay, so you just write inside lah. Okay, don't just simply write digestive system used to digest. Full stop. It doesn't really explain about anything. What just like nervous system? What is the function of nervous system? It has a lot of nerves. Full stop. It's not function. You're not going to explain anything. You are just rewriting the statement. We try to avoid something like this lah. Okay. Pencernaan. Uh, pada saiz kecil dan okay. So just try to explain what is this lah. Okay, same thing happens here. It is about cell respiration already. So you see, it matters. So you have to know what is the content inside the cell respiration. So you see, okay, the cell itself, you are going to intake glucose and oxygen to produce carbon dioxide, water, and energy. Okay, somehow some of the question is so mean. Okay, so mean until what? Until they don't give you anything. Okay, not giving you any single word inside here. You have to fill everything by your own. That's why I say you have to get familiar lah. What is happening in cell respiration, even in photosynthesis. Okay, so give the meaning of cell respiration. Wow, This one is free marks, give meaning. Everything is included here already. So you just write long. Cell respiration is converting the oxygen and glucose into carbon dioxide, water, and energy. That's all. Okay. It means converting the oxygen and glucose into carbon dioxide, water, energy. Okay, the process of converting the oxygen, okay, and glucose into carbon dioxide, water, and energy. Okay, so that's it. This one also free, cell respiration, but word equation. Word equation means what? You make it into a formula. So everything was given. Wow, so good. Wow, the marks is like spoon feed to you already. Okay, just write down lah. Oxygen plus glucose through the conversion process. Okay, you're going to produce carbon dioxide. I write the full name lah. Okay, somehow, okay, just FYI lah, not everyone knows, okay? Glucose is the simplest sugar, okay, which comes from your major source of food lah. Okay, the bread, even the corn, the tomato, the potato, okay, they all contain glucose one. Okay, so oxygen plus glucose, you're going to make it into carbon dioxide, water, and energy. Okay, actually, I know some of you are quite good in science. As you know, CO2 is carbon dioxide, H2O is water. But, okay, don't write it down in your word equation process. Okay, don't write it down in your word equation process. You write everything in terms of their name. Okay, don't write their short name, their chemical name, not chemical name, their actual name. Okay, so okay, cell organization. Hold on, let me see which question we have here. I think we can go through 3 more questions. Good. Okay. This one, cell organization. Okay, in organism. So you see, from what thing to what thing, and then the process here, you have to know lah. Okay, so I just verbally speak. What? Yeah, okay, the first one is cell. Okay, I verbally speak only, I don't write already. Okay, so from cell to tissue, from tissue to organ. Okay, very good, organ. Okay, good try, good try. So one example of tissue in human body. So muscle tissue, nerve tissue. Okay, you have different tissue here. Right, whichever tissue is possible lah. So muscle tissue is the most common one. So give one example of system in plants. So plants, okay, they have system as well. Okay, if you are not sure, right? Okay, basically you can relate one. Okay, you can relate. We have a lot of systems, So you try to imagine, what photosynthesis is counted as what? Counted as system. Photosynthesis is not counted as system. Because system in your system, you must have organ, but photosynthesis itself is not organ. It doesn't have any organ involved inside as well. So photosynthesis is a process. It is a process of the plant doing something. Okay, so when we talk about the 11 systems, right? Okay, when we talk about system, you need to have organ. So it didn't show any system for plants here, but actually you can relate one, Okay, so you just see whether the plants have this kind of system or not. Okay, the plants excretory system don't have because the plants won't pee. Don't have bone, don't have lymphatic, okay, will not digest, don't have any muscle, don't have this one, don't have skin, then don't have nerve, don't have blood, don't respiratory system. Do they have respiratory system? Yes. Okay, the plant itself will do respiration. One thing to tell you, when the question asks you, hey, based on your opinion, do you think the plant will do like respiration? Yes, plant itself will undergo respiration as well. Cell respiration and photosynthesis, they are going to do both. Okay, so don't tell the question that the plant will only do photosynthesis. It is incorrect. Okay, so this one can. Endocrine system, no. Reproductive system, yes. Why? Because the plant itself, they bear fruits and flowers. Flowers is their reproductive organ. So just write reproductive system, or you can write respiration system. Okay. So B, this one you can see got heart, got a lot of blood vessels in the body. Name the above system. Okay, so blood circulatory system. Okay, system pengangkutan darah. Okay, so one organ that makes up the system. Sangat besar. Okay, the heart is so big. Just write heart lah. Okay, so explain briefly the function of the system. So you have to bring out two messages inside. 2 points, you bring out two messages inside. Okay, the first point is carry blood. The second point is carry oxygen. Okay, so the first point you have to say, you need this thing, you need this system to carry the blood throughout your entire body. Okay, entire body. And the second thing is why to do so, because the blood itself can carry oxygen to do cell respiration. Okay, your blood will carry oxygen to do cell respiration. So this is how you explain to get 2 marks lah. Okay, three key points has to be given. Okay, to get these 2 marks. 4, the failure of the system to function properly may cause stroke. Okay, so your blood circulatory system is not function well, then you got stroke. How does stroke affect the system? So how does stroke affect the system? Okay, actually, stroke means what? This one is not in your notes as well. Okay, not in your notes, not in your textbook, just extra stuff, which should not be happening in your exam. But once, like, just in case okay, touch wood, the question come with this kind of like what is so-called out of syllabus question, then you know at least how to do. You can see we have blood supplying to brain. Okay, so how does stroke affect the system? Stroke means what? Stroke means your nervous system got affected. Okay, so your nervous system got affected, then it will affect your body blood circulatory system, because the nervous system is no longer function well already. Okay, so this is how your stroke is going to affect your system. Okay, but actually I think the question got some problem. It should be, how does the system cause stroke? Okay, it should be asked something like this because actually you see the system function fail will cause stroke. Then the question asks you, stroke, how to affect the system? Actually the question itself got some problem Okay, so basically just telling you, if your nervous system got some issue, which stroke, okay, so your body coordination cannot function well, so the organ in your blood circulatory system cannot function well as well. Okay, but it doesn't mean you will die, just causing paralysis to you. Okay, so organ X is like stomach. What is the system? Digestive system. What is the function? You absorb nutrition, you excrete. Okay, you do defecation, you pooping. Okay, you what is it called? As well as you digest the food. A lot of different points to write randomly to inside. Okay, so 2 other organs. That's why I say try to recognize all the organs inside here. Mouth can be one of the Mouth can be one of the organ. This is what is so-called pancreas, anus, large intestine, small intestine, pancreas. Just write whichever you feel is familiar. Mouth, definitely familiar kan? Okay, your anus, definitely familiar Okay, the liver, small intestine, large intestine, okay, quite familiar, right? Okay, so just write lah. Okay, so we skip to here. Question 11. What? X is stomach. Yes, X is stomach. Okay, so here, okay, name the process S and T. Okay, you analyze this one. You have to be very careful. Check your like exam paper. okay, before you submit, before you rest lah, before you consider to let go your pen. Okay, why? Because somehow a lot of people they like get confused. Plant produce oxygen. Okay, what is the process? Okay, photosynthesis it is. Okay, it happens because of the existence of chloroplast. Okay, it happens in the chloroplast. Okay, so for the animal, you're going to release carbon dioxide. So you see, the subject itself is different. When this sentence is telling that the plant is absorbing the carbon dioxide, this is photosynthesis already. But this sentence is going to tell you that the animal releases carbon dioxide. So it is actually cell respiration. Okay, cell respiration, which happens from mitochondria. Okay, so state two differences between process S and process T. So what is the difference between them? So basically, you can explain in two angle lah. Okay, we have a lot of ways to explain. Okay, the very common one is oxygen and carbon dioxide. Okay, like how? Okay, oxygen. Who produces oxygen? You produce oxygen and you consume oxygen. This is the difference. Okay, so for carbon dioxide, you produce carbon dioxide, you consume carbon dioxide, and this is the another difference. Okay, not just this, you can say, when does it happen? Cell respiration happens every time, always happening. Okay, not to say that you have 1 minute stop to do cell respiration, you will die one. Okay, so 100% anytime doing. But for photosynthesis, only will do with the presence of light. Okay. Another point what you can write here is photosynthesis is making food. Okay, but this one is making energy. Okay, cell respiration is making energy. Okay, so another one, photosynthesis itself. What is it already? I found 1.1. Wait, I lagging already. Okay, photosynthesis itself. Oh yeah. Okay, it is what is it called? It is consuming energy. Okay, from light energy. But for animal, right? Okay, it is going to produce energy. So you see the difference is here. Okay, a lot, definitely a lot one. Okay, so state two differences one. Okay, like as long as the answer is applicable, you just write inside lah. Okay, how do both processes complement each other? Complement means helping each other. Okay, need each other. So same thing happens here. 3 marks. We have to explain in three different aspects. Okay, the first aspect is oxygen. Okay, you have to say the animal will consume oxygen, which is produced by the plant. Or the question asks you process, right? Okay, process. You say photosynthesis. Okay, will produce oxygen for cell respiration. This is the first why, the first complement. The second complement is same thing lah, just change to carbon dioxide. Photosynthesis need carbon dioxide, which is produced by cell respiration. Okay. The third point is you have to like make a small conclusion already. You say, if we only left photosynthesis, all of the organism in Earth will eventually die. Okay. Vice versa. Okay, you can explain it another way lah. Okay, if now we only have cell respiration, we got no photosynthesis, eventually all the organisms in the earth will die. Okay, so this is how you explain about the processes lah. Okay, so four, predict what will happen if process S does not occur in life. Eventually, no oxygen. Okay, you cannot. Of course, you can say eventually all the animals will die lah. Okay, also can. But I'm going to the like another perspective of no oxygen because no photosynthesis. Okay, so B, this is what I say. Okay, the photosynthesis process. Why? You have to keep it in a dark place for a few days. Okay, both of the plants, before you do this experiment, actually, you have to keep them both in a dark place first. Why? Because you have to make sure both of them has no plants initially. Okay, initially comes without any starch. Only then you start to do the experiment. Okay, so two, state the constant variable. This one, the variables. Okay, I mean secondary school science is definitely different from primary lah. Okay, they'll ask you about a lot about the variation, especially when it comes to experiment. Okay, so you have to know what is constant variable, what is manipulated, what is responding. Okay, so respective here lah. Okay, like dimalarkan is constant, dimanipulasi is manipulated, bergerak balas is responding. So constant variable means the thing that is not changing, that cannot be changed in the experiment. Okay, manipulated means the thing that has to be changed in the experiment. Okay, so responding variable means the thing that you want to know in the experiment. So constant variable, type of plant. Okay, you cannot like I use broccoli here and I use cabbage here, different plant already, ma. Then the experiment is like not fair lah. Okay, so other than you can say what the size of plant. You cannot simply say I find a tree compared to a small flower, different thing, ma. Okay, so the size matters as well. So you have to make this thing constant lah. Okay, so three. Okay, this is what we so called. Okay, whether yes or no. So starch test is about iodine. Use iodine to test whether got starch or not. If got starch, yes. Okay, got photosynthesis. Where does the starch come from? Okay, photosynthesis is going to produce glucose, right? Okay, your glucose plus plus plus, okay, will slowly become starch. So if you want to know whether the plant itself got loose photosynthesis, you go and test whether got starch or not. Okay, so if got starch, it turns into blue-black color. BM is what is so-called biru tua. Okay, if no starch, then no change. No change means no photosynthesis. So the question is asking you about what? Okay, give one inference. Okay, based on the result of the experiment above. Okay, inference. Okay, just now we went through inference already, right? Okay, your observation plus explanation. Okay, so plant U does not have starch. Okay, because yellow color ma. Okay, while plant V do have starch. Okay, no need to explain whether photosynthesis or not. No, because you got 2 marks already. Okay, you just explain in one layer. You don't need to explain too deep lah. Okay, once you write wrong, the marks will be deducted. So you have to learn how to allocate the marks lor. Okay, let me think. Okay, so that's all lah. All right. So if you have any question, you can stay down to ask me. Otherwise, we call it a day lah. Okay. All the best in your revision and all the best in your first exam. Thank you for your time. See you next week. Bye bye. Okay. Thank you. Bye bye.